Today we're going to be talking about different types of neural measures, or essentially different types of brain imaging techniques. And I want to first start out by saying that some of these imaging techniques are going to tell us about brain function. So, in other words, how the brain is acting when exposed to a certain stimulus or when a person does a specific action. And others are just going to talk about, or show us rather, brain structure. And I've indicated the ones that give us information about brain function by just having the word function right here. So let's start off with talking about an EEG. In EEG, if you've ever had a sleep study done, you probably are familiar with what an EEG is like. Electrodes are placed on the scalp, and that measures electrical activity in neurons. So we're going to be learning during the course of this unit about brain cells, or individual neurons, as they're called. These neurons produce electrical activity in the brain. They produce an electrical signal. So what an EEG does is it monitors that electrical activity in different parts of the brain. So for example, symptoms of depression and anxiety correlate with increased activity in your right frontal lobe. That is a brain area that's associated with behavioral withdrawal and negative emotion. So someone with anxiety or depression, when given an EEG, may see increased electrical activity in that part of their brain. An MEG is also showing us function, but in this procedure, a head coil records magnetic fields from the brain's natural electrical currents. Again, those electrical currents coming from the activity of neurons. An example of this is that soldiers with PTSD, compared to those without PTSD, have stronger magnetic fields in their visual cortex, the part of the brain that's responsible for vision, when they view trauma-related images. So this is something that we can see when showing these types of images to people with and without PTSD. We can monitor the different levels of brain activity on an MEG. A CT scan is essentially just an x-ray of the head, and that'll generate images that might locate areas of brain damage or injury if someone's been in an accident. A CT scan is going to show us brain structure, but not nearly to the same level of detail as an MRI. So children's brain injuries shown in CT scans can predict or correlate with impairments in their intelligence and in their memory processing. Again, this is not going to give us the best picture of the brain structure. We're going to get that from doing an MRI. So why don't we talk about MRIs while we're at it? This is also going to tell us, maybe some of you have had an MRI before. I've had an MRI before. You're put into like a kind of a very, very loud tunnel. And I've had an MRI of my brain, which if we were in school, I would show you my brain scans. We'll have to wait till we're back in school. But essentially, they put on your head what kind of resembles like a catcher's mask, but this device goes all around your head to keep your head and your neck completely still. You lie down in the chamber, and it uses magnetic fields and radio waves to produce a map of the brain structure. So the reason why magnets are able to detect blood flow in the brain, using a functional MRI that is because your blood has iron in it. So a functional MRI, similar to an MRI, but this is actually going to measure blood flow to brain regions by comparing continuous MRI scans. So just think of like a flip book, like these MRI scans are taken continuously, so that when they're put together, we get a sense of the activity in the brain by looking at the changes in blood flow. The example for this one is that after surviving a near plane crash, passengers who viewed material related to their trauma showed greater activation in areas of the brain that are involved in fear and memory and the visual centers than when they watched footage related to the 9/11 attacks. Coming back to, a PET scan. A PET scan is also showing us brain function, but instead of tracking blood flow, it's tracking a radioactive form of glucose that is injected into the person before they go into the scanner. So what that's essentially doing, it's tracking where this radioactive glucose is being metabolized in parts of the brain that are most active. In the parts of the brain that are active, those will be the areas where we see the glucose being metabolized. So if you put someone into, or if you were to give someone a PET scan, and have them perform a specific task. The PET scan is going to show the part of the brain that's active by tracking the metabolism of this radioactive glucose. An example of this, monkeys with an anxious temperament have brains that use more glucose in regions that are related to fear and memory and expectations of reward and punishment. So that's a little overview of these different types of brain scans. We're going to see an fMRI used in a video that we watched called The Love Competition. Lesioning is another technique. In lesioning, what happens is a part of the brain is purposefully, destroyed in order to observe differences in behavior. This is not something that is done in humans. But I have a picture over here for you showing you that Carl Lashley did this with the brains of rats, searching for what is called the engram, or the physical trace of memory in the brain. So what he did was, in creating these lesioned areas, was trying to see if he lesioned a particular area, would the rat lose its ability to remember, perhaps amazed that it had learned the day before. But what he found is that regardless of the part of the brain that was lesioned, the rats did not lose their memory for what they had previously learned. And that tells us that there is no one place in particular in the brain where memories are stored. They're stored likely in networks of neurons throughout our cerebral cortex, or throughout the outer layer of the brain, where our higher level thinking occurs. Now, in humans, though, we learn some of what we know about the brain from people that have had suffered brain damage to different areas, that have had tissue damage in different areas of the brain. And so we can learn quite a bit, not from purposefully damaging their brains, but from people that have experienced brain damage of some kind. So this is just showing you again what an EEG would look like. You see an individual with electrodes placed on their scalp that is measuring the electrical activity, or the activity of neurons in their brain. This is showing you a CT scan versus an MRI. You could see much greater detail in an MRI compared to a CT scan, which is more like just an X-ray. And a PET scan will produce an image that looks something like this. Areas of the brain that are metabolizing the greatest amount of that radioactive glucose are going to appear in red on a PET scan. PET scans are used for people who have cancer. Cancer cells metabolize that glucose at a very fast rate, and so that's why PET scans are used to, in order to locate areas of the body that may have cancer cells. Other really interesting techniques that are used, and these are not actually ones that you need to know about for the AP test necessarily, are an ECOG. Think of an ECOG as similar to an EEG, where you're measuring the electrical activity in the brain, but you're bypassing the scalp and actually implanting the electrode directly onto the brain and measuring the electrical activity in the brain that way. Another very interesting technique is called the Utah electrode, or a multi-unit recording. So you're seeing that working here with an individual who is blind. A way that some scientists have been working on helping blind people to see is by implanting one of these Utah electrode arrays into the visual cortex. The person then wears glasses with a camera. So he's looking at an image. That image is being picked up by the camera, and the on the glasses, that is. And the camera on the glasses is actually sending a wireless signal to these implanted electrodes in the occipital lobe, or in the visual cortex, that is enabling the person to see. Because as we're going to learn in our next unit, we don't see with our eyes, we really see with our brain. All visual processing, the information comes in through our eyes, but the visual processing itself takes place back here in the visual cortex. And a last really interesting technique to know about is called optogenetics. So this visual right here is going to explain to us how optogenetics works. But essentially, neuroscientists are starting to specialize in studying and controlling individual neurons, or individual brain cells, by using light. So in the first step, scientists use gene therapy to encapsulate genetic material into a virus, essentially, that's going to be used to transport genetic sequences into living cells. That genetic material in the virus is designed to encode itself in cells with traits of a light sensitive protein called opsin. So essentially, implanting this virus or inserting this virus is going to make a specific group of cells sensitive to light. So the virus is delivered usually through an injection into targeted cells or targeted neurons in the brain that are going to turn that genetic sequence, or transduce, I should say, the genetic sequence of the opsin, making those cells now sensitive to light. Then using fiber optics, using something like some kind of light source, they're going to actually emit a signal to those individual brain cells or to those individual neurons, again, via the light source. Those neurons will then communicate the signal in a chain reaction to other neurons, and that is going to actually produce an action in the organism, in this case, in the rat. So it is very, very interesting the way that scientists are now able to use light to control the activity of individual neurons. We will talk a little bit more about brain-computer interfaces, but at the heart of how a lot of these brain-computer interfaces are working is with these electrodes that have been implanted. So these tiny little implanted electrodes can give scientists not only a window into how individual brain cells or neurons are responding to certain stimuli, but like you see here in this case, they can also give them a way to actually isolate those specific brain cells or those specific neurons in order to produce a response in the individual. In this case, in order to enable them to see, or in some cases, enable them to literally think about an action and carry out that action via a prosthetic limb. So really, really cool stuff that we'll talk more about.